So good morning once again, and we're going to be finishing up lecture 2 today. Maybe we'll have some time, towards the end to do a little bit of review. Maybe not, I'm not sure. We'll see how it goes. But then your quiz is going to be next Thursday. I will tell you more about the quiz later when more people show up. All right, so let's begin with these two questions that we haven't finished from last class, and these are questions related to carbohydrates. So that's the first one right here. Sucrose is made up of which of the following sugars? So just to refresh your memory, we have glucose, which is a monosaccharide. Monosaccharide means single sugar, right? And you can combine it with another glucose, right? And that's going to give you maltose. Right? This is the disaccharide, double sugar. You can also take glucose and you can combine it with fructose, right? And that is going to give you the sucrose, which is your kitchen sugar. And the last one is pretty straightforward. You have glucose plus galactose. When you see the word lactose, right, in galactose, you know it is going to give you lactose. So all these are disaccharides, and you can get them by combining monosaccharide. Does anybody remember the name of the process that we use to connect the two monomers together? What's the process? Okay, Natha said hydrolysis, but that is the opposite. Okay, hydrolysis is to break down polymers. If we were trying to join them together, we need to remove a water molecule. What is the name of that process? Dehydration, that's right. We have to do dehydration synthesis. This is not really the study guide, medicine. This is just like, I'm taking up the question is all. So that's right. So these are dehydration synthesis reaction. Okay, and each of them involves removing a water molecule and connecting monomers together, forming a bond, right? And these are all covalent bonds that we're talking about. So the correct answer for this question would be D. Glucose plus fructose. All right, moving on. Now, this is a little bit more involved. Let me give you a quick review before we do this question. We learned about four types of polysaccharides. Okay, polysaccharides are really, really long chains of carbohydrates, and you basically connect multiple glucose molecules together to form a long chain, right? So we learned two types that are useful for storing energy, and we learned two types that are useful for forming structure, right? Structural support, right? So we have the glycogen and starch. These are the storage form of energy. Right? For glycogen, it is storage form of energy in animals. So we store them in muscle, we store them in our liver, right? And when we have excess glucose, that's where we will put them. For starch, it is the storage form of energy in plants. Okay, they do photosynthesis, they make food, and then they store these excess food in the form of starch in stems and roots, right? So these are for storage of energy. On the other hand, we have cellulose and chitin. Okay, these are for structural support, right? Cellulose forms the fibers in plants, and right? Form the cell wall around the cell, gives the crunchy part, makes up the crunchy part of the plant, right? Chitin is the specialized polysaccharide that forms the shell of crabs, lobster, right? clams, and so on and so forth. They also form the exoskeleton of insects, right? So these are the 4 kinds of polysaccharides that we have learned from last class. Keeping that in mind, let's take a look at this question. This will be an example of a multi-select question, okay, where you will have to choose all the correct answers, not just one. So I'll give you a moment to read that through and I'll take it up. I will launch the poll for you to see, to try. Go ahead, please.
I'm sorry, what are we supposed to be selecting? Because I only see question 11 on the study guide and I'm kind of confused.
Question, oh, this is not the study guide though, right? This is the PowerPoint slides that from your notes.
Yeah, but what she's saying, we're seeing question 11 where it's the chart.
Yeah, we're not seeing the slides.
You're not seeing the slide?
We're seeing
No.
chart.
Oh my goodness, I'm so sorry. That's why you guys are, oh, okay. Sorry. Oh, people who are watching this on YouTube, you're probably thinking what is going on. I forgot to share the right screen, that's all. Sorry, thank you for letting me know. So this was the previous question. I was writing all these things, right? No wonder, like, you guys are a little bit lost. Sorry. Okay. So I was just reviewing the different combinations that can give you the different polysaccharides. Okay. Now we're doing these questions. Okay. So I was saying glycogen and starch, that makes up the storage form of energy, and cellulose and chitin, that makes up the structural support. Are you guys okay now? Can you see my screen, or am I still at the wrong thing?
Now it's good.
Okay, thank you so much. Sorry, guys. It's one of those days. So let me relaunch the poll for you. Okay, I'm gonna leave it at that. Thank you for those of you who have selected something. Let me share the results with you. So this is an example of a multi-select questions. All the multi-select questions on the quiz will tell you explicitly to select all correct answers. Okay, so you will know whether it's a regular question or multi-select question. And for the regular question, you wouldn't be able to choose more than one answer anyways. Like the system will not let you do that. So here, let's take a look at the first one. Polysaccharides are important for both energy storage and to provide structure to cells. That is correct, right? Glycogen and starch, that's for energy storage. Cellulose, that would be providing structure for cells, right? Forming the cell wall. So that's a correct statement. We're looking for the false one. Number 2, glycogen is important for both energy storage and to provide structure for cell. That is incorrect. That's the false statement because they do not provide structure for cells, right? That's the job of cellulose. Next one, cellulose is only found in animal. That is incorrect. We do not have cellulose in animals, right? We don't have cellulose. I mean, we can eat them, but like we don't produce them, right? Cellulose are found in plants. Glucose, fructose, and galactose are examples of monosaccharides. Monosaccharides are the single sugar, the monomers. That would be the correct statement. And finally, starch is a storage polysaccharide made by plant cells. That is also correct. So the incorrect statements are B and C. All right, now we're going to move on to lipids. Lipids is our next macromolecules. And it could get a little bit confusing with all the slides that I'm going to show you. So let me just give you a quick summary here first, and you can write it down if you want. Okay? Lipids, we will be looking at 4 types of lipids. Okay, 4 types of lipids. Okay, first one is fat, okay, which is also known as triglyceride. I'll explain what that means later. And under fat, there are 2 kinds of fat. We have saturated fat and unsaturated fat. Okay, you don't have to know what they mean right now. I'm just showing you an overview. So when we go through the slides, you can see where we are at. The second type of lipids we will look at is called phospholipids. Phospholipids, and there are no subcategories for phospholipids. And we will spend 1 slide on wax. And finally, we have steroids. Okay. So those are the 4 types of lipids that we will be looking at in the upcoming slides. Right. So give you a moment to write down what you need to, and we'll go on from there. Lipids. Just like carbohydrates, lipids are composed of carbon, hydrogen, and oxygen primarily. Okay, there are some cases where you would have a little bit of a modification, but mostly it's carbon, hydrogen, and oxygen. Now, it's a little bit different when it comes to lipids, right? Compared to the other 3 types, compared to carbohydrates, compared to proteins and nucleic acid. Lipids is a little bit different because it does not contain long chains of repeating units like the other 3. Okay? You know how in polysaccharide, it's multiple glucose all linked together to form this long train, right? Kind of like a GO train, right? Okay, this is not like that. Lipids instead will have different components that we put together to build the lipids. It will become more clear in a bit. But for example, let's say there are 3 components. Okay, let's say there's A, block B, and block C. Okay, so to build, let's say to build fat, you will need to take 1 of A and then 3 of B's. And that's it. You're not gonna have multiple B's all linked together, right? That would be like polysaccharide, right? If let's say we want to build steroids, then I would need to take 4 of C's and put it together. Okay, so that's the main difference. We're not trying to connect a long chain of stuff together. We're just, picking and choosing what we need, and then we put it into 1 molecule. Okay, so that's the main difference. Okay, we are joining components together rather than long chains of monomers. All lipids are going to be hydrophobic. Can someone explain to me what that means? What is the meaning of hydrophobic? We learned that last class. Okay, no water resists water. That's right. Hydrophobic means it is not going to dissolve in water. Okay, it is not going to dissolve in water. Right? It can sometimes form a water-resistant barrier. Okay, so they are all hydrophobic. This is a list of functions of lipids in general, okay? And then I'll tell you which one does what later on. In general, lipids are good for storing energy. Okay. Now I know what you're thinking. You're like, hey, isn't that the job of like glycogen, right? Or like starch? Yeah, but then those are for short-term storage. So let's say you skip breakfast this morning, right? Your body will be using the glycogen reserve from your muscle or from your liver, right? It will take the glucose from those places and allow you to burn it for energy. But if you are fasting for a couple of days, okay, or if you're stranded on an island and you don't have food for a couple of days, you're gonna start to run out of glycogen to burn. So you would switch to lipids, right? So lipid is for long-term storage, long-term. As you will see, lipid is also a main component of cell membrane. A cell membrane it's like a barrier that surrounds each cell, and it's important to have this barrier to help regulate, to help control what goes in and goes out of the cell. Okay? And lipid is a main ingredient for that cell membrane. And they can also form water-resistant barriers. Okay, Nathan already mentioned that earlier. So if you think about like the skin of an apple, right, that's water-resistant, right? Like our skin is water-resistant. Like we go into the bathtub, we don't like suck up all the water, right? We're not like sponges, right? Like the water will repel, right? So we have a water-resistant barrier in the skin. This picture here shows what fat cells look like in the body. And all these white spaces, those are basically lipids that are being stored for long-term energy storage. So those are the generic functions of lipids. Okay, and then as we go on, I will tell you, like, which one of these does what. Okay. So where do we get fats, right, in general? Fats are found in most of our food sources. I mean, like, it's really weird that people would deliberately, find fats to consume in their diet, right? It's just part of the food that you eat. You don't have to purposely find them. Okay, you will find fat in, in most meat product, in all meat product, I would say. Okay, although some are more rich in fats than other, right? Like a salmon, right? Salmon is pretty fatty. There are vegetables and nuts that are high in fat content. And if you really, really want to just like consume fat on its own, then we have the butter and oil, margarine. All those things are really, really high in fat content. When it comes to fat consumption, there are certain kinds of fat that we need in the body, but we are not able to make them. Okay, so these are called essential fatty acids. Essential means you can't make it in the body. You cannot make it in the body. You must eat it. Okay, and these are required for normal body function. Example of that would be omega-3. You probably have also heard of omega-6 and omega-9, right? So these are some examples of essential fatty acid. Okay, so omega-3 is not one thing. Okay, there are many different kinds of omega-3. Right, ones that you might be familiar with, especially if you if you have kids or you're pregnant before, then your doctor would tell you to take supplements of EPA and DHA. They are omega-3s that are vital for healthy brain development of the baby. So these are naturally occurring in foods. You don't really have to take supplements for it, but some people take it because it's easier, right? So you will find them in things like avocado, salmon, eggs, right? So let's take a look at the structure of fats. Okay, remember I wrote it down for you, right? There are 4 types of lipids, right? Fat, phospholipid, wax, and steroids. We're starting with the first one, fat. Fats is also called triglyceride. If you do a body check, your doctor will check your blood cholesterol level. That's the one that people are familiar with, but they will also check your triglyceride level. Okay. And that's basically what it is. Triglyceride is the fat. What components makes up the fat? Well, we have this molecule called glycerol. Glycerol. Okay, sometimes people call it the ho molecule. You see HO. You don't have to know how to draw it. Okay, you don't have to recognize it. But I'm just telling you, this thing is called glycerol. And we are going to need to have 3 fatty acids. Okay, fatty acids are just a bunch of C and H connected together. Sometimes they are really short, like the ones that are being shown here is only has 5 carbons. Sometimes they are really long, okay? 10s, 12s of carbons, right? The length doesn't really matter. Okay? So you will have one glycerol, and then you will attach different fatty acids onto it. Okay, you will attach up to 3 fatty acids onto the glycerol to get the fat molecule. So how do we connect the fatty acids to the glycerol? It's the same process that I have been telling you since the beginning of the lecture, right? It's the dehydration synthesis, the dehydration reaction. You would remove an H from the glycerol and you will remove the OH from the fatty acid, and that creates 1 water. But I had to do it 3 times. So remove water, remove water, remove water. You will create 3 separate connections, 3 bonds that holds it together, and you would have 3 water molecules that comes out in the end. Okay, so what do we need fat for? Fat is, like I said, excellent for energy storage. Okay? But they usually will form around major organs as well. So around your heart, around your kidney, liver, it's common, it's actually normal to have a little bit of fat that surrounds it. The fatty tissue will help cushion the organ as well as help insulate them. To insulate them means to keep them warm, right? When it's minus 40 degrees outside, you don't want your liver to freeze, right? So those fats will keep it nice and warm. So that is the triglyceride. Does anybody have any questions so far? Great. Now, we will take a look at the kind of fatty acids that is being attached onto the glycerol. If you were paying attention earlier, when I roll lipids, and then I have fat, under fats, there are 2 categories, right? Saturated fat and unsaturated fat. So which one do you think is healthier? If you gotta chuck down some fats, right? Okay, like would you rather have more saturated fat or more unsaturated fat? Which is better for you? Okay, I have a few people, 2 people said unsaturated fat. What about does anybody think it's the other way around? You guys agree? Okay, so that's correct. Unsaturated fat is the better one. Okay, you can have more of those and it will be okay. Saturated fat is the one that you try to keep it at bay. Okay, you don't want to have too much of that. And here's why. All right, so saturated fats have the glycerol, just like the unsaturated fat. But what sets them apart is the tail that is being attached onto it. It is the kind of fatty acid that is attached to it. Okay, it's called saturated because the carbon atoms in the fatty acids are bound to a maximum number of hydrogen atoms, meaning they are full of hydrogens. You can't put any more hydrogen on them anymore. They are saturated with hydrogen. That's where the name comes from. Now, when that is the case, the tails tends to be relatively straight. So let's say I draw a saturated fat, right? This is the glycerol. Let me try that again. This is the glycerol. And then you would have the tail. 1 fatty acid, 2 fatty acids, 3 fatty acids. Those are saturated fatty acids. They are bound to as many hydrogen as possible. So let's say I draw another saturated fat molecule next to it, right? And then I can draw another one here like this. Now, because the tails are straight, you can pack them pretty closely to each other, right? They're not going to get in each other's way. Imagine like lining up at the front of a classroom, and everybody has their hands straight down, right? Like you can stand really close to your classmate and not have a problem, right? And that's exactly what the saturated fat is doing. They can pack really closely, and they tend to be solid at room temperature. Now, here is the problem. If they can pack closely together easily outside of the body, they could also pack quite easily inside your body, right? So if you consume excess amount of saturated fat, that's exactly what they would do. They would start to build up in your arteries, right? They would start to cause the arteries to become hardened, right? And you might start clogging it over time, right? So you want to avoid saturated fat. Most animal-derived fats are saturated, okay? So butter, lard, right? Some people like to use lard to make like pastry, right? It's really tasty, but not very healthy. Okay, bacon grease, right? All these things are saturated fat. All right. Now the opposite of saturated fat is unsaturated fat. Okay, listen carefully. Unsaturated fat also has the glycerol, and then at least one of the fatty acid is going to be an unsaturated fatty acid. Okay, so in this picture, right, you can see that's a saturated fatty acid, but the third one here, that's an unsaturated fatty acid. Okay, so you have to have at least one unsaturated fatty acids. You can have two or you can have three. The more you have on the molecule, the healthier it's going to be. Okay, so if all three tails are unsaturated, then we call it a polyunsaturated fat. Okay, that's supposed to be like the healthiest of different kinds of fat. Okay, so what is an unsaturated fatty acid? Well, they contain a double bond. For those of you who haven't really done chemistry before, don't worry too much about what a double bond is. Okay, it looks like this. Look, normally a carbon would connect to another carbon with one line. The line represents sharing of electron, right? We talked about that last time. If you have a double bond, it just means you have two lines. You are sharing more electrons. That's all it means. Okay, instead of sharing one pair of electrons, now you're sharing two pairs of electrons. The key thing to know in the context of this is when you have the unsaturated fatty acid, it makes the tail bend like that. Okay, so now if I try to put another unsaturated fat next to it, like it can't get really close because then the kink here in the tail, it's kind of getting into the way, right? So imagine lining up again, but then like you're sticking your arm out, you're sticking your feet out, and people can't really get close to you, right? So if the molecules are not getting really close to each other, then they would not pack into a solid, right? So for that reason, unsaturated fat tends to be liquid at room temperature. Okay, so they don't pack as easily in the body as well, which is why they're a little bit healthier. Most plant-derived fats are unsaturated. Okay, so olive oil would be an example, right? Any other oil that is unsaturated that you can think of? What do you guys cook with, huh?
Coconut oil.
Okay, Hiba, you actually read my mind, sort of. I wanted to talk about coconut oil as an exception, actually. Okay, coconut oil, even though it's plant derived, but it's solid, right? Like it's a white paste, right? Yeah, it's not really like you don't really pour it, right? I mean, there might be a little bit of liquid at the top, but for the most part, it's like a white paste, right? Am I correct in saying that? So that is an exception. Okay, so that's why here it says most plant-derived fats are unsaturated. Okay, but coconut is an exception. That one is actually saturated. What else? Come on, give me something else, guys. Like, I don't think you guys all cook with olive oil, right? It's Francis says it depends. Yeah. So like, what else? Give me other examples.
Like vegetable oil, canola oil?
Yeah, canola oil, vegetable oil, avocado oil. Excellent, right? Okay, so anything that you can pour, basically, is going to be unsaturated fat. Okay, peanut oil, grapeseed oil, okay? You name it. Sesame oil, excellent. Those are very good. I love using sesame oil. So that's it. Okay, do you guys understand? Saturated versus unsaturated. Okay, they both have glycerol. What separates them is the tail that you attach onto the glycerol. Okay. Earlier, I was telling you about essential fatty acids. If you forgot already, essential fatty acids are fatty acids that we need in the body, but we cannot make it. Example would be omega-3. Why is it called omega-3? You don't really have to know this. I just tell you because it's, sometimes it's good to know why things are the way they are, right? So this is the one of the fatty acids, okay, in the fat. And the extra line represent a double bond, right? So where the first double bond occur will determine what do we call it. So this is carbon number 1, 2, 3. First double bond happens in carbon 3. That's why it's called omega-3. If the first double bond happens in carbon number 6, then we say omega-6. Okay? That's what it means. And so that's EPA, that's DHA. Both are important for brain development, and they are good for your heart as well. Okay. So some people take them regularly as supplements. Now, I don't have time to go through this, but very, very quickly, there is like a little video clip that you can watch. People buy organic eggs. People like to buy organic food nowadays, right? Because they think it's healthier, because they think is, sometimes better for the environment. And one of the reasons is people also think they are more nutritious, okay? So eggs are expensive nowadays. Like if you buy a dozen of eggs used to be like $1.99, right? And now a dozen of eggs is like, $2.99 and it's already like on sale, right? Okay. But if you buy organic eggs, which I don't, but some people do, organic eggs is like super expensive. We're talking about like $7.49, right? For a dozen, right? And some people buy it because they think it has more omega-3 in it, it has more omega-6, and it's more nutritious. But is that true? Is that true? Okay. So if you're interested, you can watch this clip. It's a show on CBC. Some of you might have heard of it before. It's called Marketplace. And then they actually got eggs from a wide variety of sources, from farm, from different brands, and then they check the nutritional value to see, if organic eggs are actually more nutritious. Should you be shelling out the extra money to buy those organic eggs? Spoiler alert, probably not, okay? Okay, moving along. We have another slide here. Take a look at the picture. Okay, one of them is saturated and one of them is unsaturated. Okay, I'm gonna launch a poll, and you tell me which one is saturated. We have A on the left and B on the right. I'm asking which one is saturated. Okay, very good. Most of you got it right. It is the one on the left. What gives it away? How do you know that one is saturated? Okay, tell me, what's the reason, huh? No tail. Not so much no tail. I mean, all these these are the tails, right? These are the tails. Okay, and as are these. That's right. No curve, right? Okay, you don't see a kink. You don't see a bend in the tail. That's why you know this is saturated and that's unsaturated, right? Okay, so these kinks, okay, these bends is as a result of the double bond. You see the double bond here, double bond here, double bond, double bond, double bond. Right? Remember, only one tail has to have the double bond for it to be considered as an unsaturated fat. All right, trans fat. So even though unsaturated fats are healthier, but it's weird to like pour olive oil onto your toast in the morning, right? Okay, or pour like avocado oil onto your toast, right? Like it just doesn't feel right. So people came up with this idea. You know what? Let's start with the unsaturated fats. Oh, and Jelly has a question, or sorry, Angel Edward. Yeah, please. What's the question?
So is it the fact that it's like closely packed together make it saturated, or?
Oh, so what makes it saturated is that the tail does not have a double bond.
Oh, okay.
And because it doesn't have a double bond, it allows like two different molecules to be able to pack together closely. Does that answer your question?
Yeah, okay, thank you.
Okay, great. So people had this great idea. They're like, you know what, let's start with the unsaturated fat. And we will use a chemical process to add more hydrogen to it so that it becomes a solid. Okay, remember, unsaturated fat means it still has room for more hydrogen, right? So in a chemical process called hydrogenation, you put these oil under pressure and then you add hydrogen atoms to it, and then it turns it into a solid. Okay, not like a chunk of solid, but then like a semi-solid. Okay, like margarine, like shortening. That's what they are. They are hydrogenated vegetable oils. Okay, and that used to be okay. How should I phrase this? It's kind of bad. Okay, because the process creates a byproduct called trans fat. Okay, our body does not have the enzyme to break down trans fat. So trans fat just kind of builds up in the body over time, and it contributes to things like cardiovascular disease. It contributes to diabetes, right? And so it's like kind of like the worst kind of fat to have in the body. It used to be a lot of fats, a lot of food has trans fat in it. Okay, especially like fast food, or if you buy snacks from the shelf, there's a lot of trans fat in it because the trans fat makes the food taste better. Like if you do your fried chicken with some trans fat in it, it tastes better. If you use it for your product, it gives them a longer shelf life. So trans fat was actually pretty ubiquitous. It's kind of everywhere. But then people are becoming more aware of the negative health effect of trans fat, and then they started to become more vocal, right? And finally, in 2018, Canada, we officially banned trans fat. Okay, so there are no more trans fat allowed in fast food, no more trans fat allowed on anything that's sold as a food product. Okay, so now when they make the margarine, they actually have to make sure they remove the trans fat from the product. Okay, to make it healthier, I guess. Any question about anything that I talked about so far? Okay, let's move on. Does it taste that different? You know what? I actually never did a taste test. Like I don't have one that has that's made in trans fat and made in not, and then able to tell the difference or not. But supposedly it makes them taste better. For me, like fried chicken is fried chicken, right? It can't be that different. All right, next, we're gonna talk about lipids, another kind of lipids. This is called phospholipids. All right, so phospholipid has three components. All right, we have one glycerol. You should write this down. One glycerol, just like before. Two fatty acids. Now, these fatty acids can be saturated, can be unsaturated. It doesn't really matter. And we need to have a phosphate group, phosphate head. Okay, sometimes it's called polar head. Okay, so in this picture, you see the polar head, and then you kind of see the glycerol right here in the middle. You don't have to identify them, I'm just telling you. And then you have your fatty acids down here. Okay, so for phospholipids, it has the carbon, it has the hydrogen, it has the oxygen, but then it also has the phosphorus, okay, phosphorus, because of the polar head. Now, what does it mean, like polar head, non-polar tail? What does it all mean, right? So we usually draw a phospholipid like this, right? Like this. So the circle part represents the polar head. If you remember what I taught you last class, if something is polar, then it is hydrophilic, right? Hydrophilic, that means it loves water. And then the tail the tails are hydrophobic. So we have an interesting molecule here. One part of it is hydrophilic, another part of it is hydrophobic. Molecules like these are very useful, okay? Because it would allow two things that normally don't mix together to come together. Okay? Detergent is an example of a molecule that has this property. Right? If you try to wash the greasy dishes with just water, it will not get cleaned, right? Okay. But if you add a little bit of detergent, then you can wash off the grease. And the reason is because the detergent has a hydrophobic part that binds to the grease and a hydrophilic part that binds to the water. So it allows the water to carry the grease off the plate and, wash it away. Okay. So in the body, we need phospholipids to form the cell membrane. Okay, so if this is a cell, okay, the membrane is what surrounds the cell. And if you take a look at the membrane, it would be like this. This is outside of the cell, this is inside of the cell. So you would have two layers of phospholipids. Okay, the heads would be facing either the outside or the inside, both of which would be a water environment. And you have a thick layer of hydrophobic tails that are sandwiched in between. Right? So we will talk more about the cell membrane in another class. But for now, just know that the phospholipid forms this barrier, so things cannot come in and go out easily without without some kind of regulation. Right? That's all for phospholipids. Just stop me if you have a question, please.
I have a quick question about phospholipids.
Sure, please go ahead.
So phospholipids have like the bit of a kink, right? Like the bit of a kind of a curved tail.
The tail, yeah, okay, sure.
So it does that, right? Like that's how you can identify it, or
I mean the kink could be there, it might not be there. It depends on what kind of fatty acid it is, right? So like you can have phospholipid that looks like this, and then you can have phospholipid that has kink in it. You can also have phospholipids that has like two kinks in it. Okay. That actually is important for another reason that we'll discuss later. But don't identify it based on like whether it has a kink or not, right? So like if I have glycerol and 3 fatty acids, then I know this is a fat molecule. This is a triglyceride. But if I only see 2 tails, whether it has kink or not, then I know I'm talking about like a phospholipid, right? Does that answer your question?
yes, thank you.
Okay, great. All right, next one is wax. Don't worry too much about wax, okay? I'm just gonna spend one slide on it. It's pretty self-explanatory. Wax forms a water repellent surface, okay, basically. So like the surface of plants, surface of your fruits, they are like they have a thin layer of waxy substance. Okay, that's why in the morning you might see droplets of water that are formed on the plant. They don't like soak through the plant because of the wax. Okay, beeswax. Some people use beeswax for cosmetics, right? For like lip balm and stuff like that. And we also have waxy substance in our hair, in our skin to form water repelling surfaces, right? Okay, so they have long fatty acid tails bond to long chains of alcohols or carbon rings. Okay, so don't worry too much about that. I don't think I have any questions on the test regarding wax. Okay, just know that it is an example of lipids. Next, we're gonna move on to steroids. When people hear the word steroids, what comes to your mind? Okay, I mean, you don't have to tell me. You can just think about the thing that comes to your mind right now when you hear steroids, right? A lot of people associate with athletes, right? Who are using steroids for doping, right? Making them stronger, building muscle, right? But that's not necessarily true, right? The steroid is actually very important for normal body function, right? To make a steroid, to identify a steroid, you will see that there are 4 carbon rings that are fused together. So that's one ring. These hexagons, they're carbon rings, 2 rings, 3 rings, 4 rings. So the rings are fused together and they would have things that are attached to the rings. You don't really have to know what they mean, okay? Those things that are attached onto the ring, they are called functional groups. Okay, and you can put different functional groups onto them. That gives them different properties. Think of them as like accessory, add-ons for like your computer. Okay, like you can add like a cooling station to cool down the hard drive. You can add like LEDs to make it look pretty. So you can add different things to the core rings, the core carbon rings, and it will give the steroid different properties. Okay, you will learn more about functional groups in your second semester chemistry class. So there are different kinds of steroids in the body, and a lot of them actually uses cholesterol as a starting material to make. Okay, so people also think cholesterol is bad, right? Because the doctors say, oh, your cholesterol is too high, but actually there is good cholesterol and bad cholesterol. Okay, so the good cholesterol, we need them for cell membrane. Okay, so I know cell membrane has phospholipids in them, yes, but it also has cholesterol in them as well. Okay, and we also need to have some steroids in various other locations in the body. Bile is anybody know where you find bile in the body? Bile. Okay, stomach. Good try. Not stomach. Anyone else? Liver. Yes, Justine. The liver makes bile, and we store it in the gallbladder. Okay, and we need the bile for breaking down fat. Okay, if you have a greasy meal, you need the bile to help break it down. And within that bile, you will have a little bit of cholesterol. If you don't know what bile is, okay, it's the like you might have this experience. Like think of one time where you got like severely food poisoned or something, like you ate something really bad, and then you keep on vomiting, right? You vomit all the food out, okay? But then you're still vomiting, but there's no food left. So the next thing that comes out is this, greenish, yellowish thing that tastes really, really bitter. Okay, if you had that experience, that's the bile that's coming out. Okay, and normally they are stored in the gallbladder. Medicine asks, what if there is no gallbladder? Yeah, so some people can have to remove the gallbladder because of gallstone, and they can still make it. Okay, they just can't store it. The liver will still make it, and then you will not be able to store it. You will have to make it on demand, so you have to cut down on lipids in your diet. So no greasy food, right? No more KFC. All right, moving on. You also need cholesterol steroids to make things like vitamin D, right? Of course, you need to be in the sunlight as well to do that. And then the cholesterol is also important because it's a raw material to make steroid hormones. Okay, hormones, and please write this down. Okay, hormones are molecules in the body that is used as messengers, communicators. Okay, you have to secrete hormones to tell other body parts what to do. And there are two kinds of hormone, right? Some are protein-based hormones, which we'll look at. Some are steroid hormones. Okay, example of steroid hormones are the sex hormone. So in male, the dominant steroid hormone is testosterone. Right? To make that, you need to have cholesterol. Right? So you see the ring, right? 1, 2, 3, 4 rings, and then you have these functional groups that are attached to it. Okay. For female, again, you have the 1, 2, 3, 4 rings, and then different functional group attached to it. The one is in female is estrogen, right? What's the other one called? Huh? What's the other female hormone besides estrogen? That's right, progesterone. Excellent, Nina and a few other people. Right, so we need to make these hormones, right? This, give us our identity, right? So cholesterol is not all bad. That's my point. All right. All right, let's do some poll questions, and then we're gonna move on to proteins. Here we go. All right, anyone else? We are at the 43-second mark. I'm going to close the poll when it reaches a minute. Is it because you guys don't see the poll? Is that why you guys not choosing anything? I'm just curious. All right, that's fine. Here we go. Glycerol combines with 3 fatty acids to form what? Okay, the correct answer is triglyceride, right? Tri means 3. Like triangle has 3 sides, right? Tricycle has 3 wheels. Triglyceride. There are 3 fatty acids. Okay, some people chose A. A is not correct. A is, write down if you didn't know. It's glycerol plus 2 fatty acids and a phosphate group, right? Okay, so that's phospholipids. Trans fat is what you get when you do hydrogenation. It has nothing to do with what the question is asking. That's it. Oh no, that's not it. One more. Which of the following is false about lipids?
I'm sorry, I didn't get to write down the last part. Can you go back one slide?
Sure, there you go.
Thank you.
Phosphate group, yeah, phosphate group.
Okay, thank you.
Okay. You good now?
Yes, thank you.
All right. Which of the following is false about lipids? Okay. Thank you. The answer is kind of all over the place. Am I going too fast, guys? Like, is it why you are not absorbing the information? Am I talking too fast? What's is there any concern that I should be aware of? Because I I would expect you guys to get this question right, honestly. No? Okay. So false statement, right? Let's see. Steroid has at least 1 fatty acid tail. That's the false one. Steroid. Remember steroids? Here. The only thing that makes it a steroid is not whether it has fatty acid tail or not, right? Like to know whether it's a steroid or not, what do we look for? We look for the 4 rings, right? Remember? That's the only thing. Other stuff that you attach on, those are just functional group, and it doesn't have to be a fatty acid tail, right? So that's why this is false, right? It says it has to have at least 1 fatty acid tail. Not true. Everything else is true, okay? Canola oil, which is a liquid, contains unsaturated fatty acid. That's true, right? That's why it's liquid, right? At room temperature. Triglyceride is fat and is long-term storage of energy. That is correct. Cholesterol is a steroid. Look, it literally says right here, right? Steroid, cholesterol, right? Cholesterol is a base steroid that we use as raw material to make other things, other steroids in the body, including hormones. Glycerol is a component of triglyceride and phospholipids. That is correct, right? Triglyceride is 1 glycerol plus 3 fatty acids. Phospholipid is 1 glycerol plus 2 fatty acids plus 1 phosphate group.
I'm sorry, I have a quick question.
Sure, please.
How would you make the false statement into a true statement?
So we would say steroid has I'm going to cross this all out, has 4 fused carbon rings. Okay, that's what I would say.
Okay, thank you.
Yeah. Anyone else? Like, we together? Give me some, feedback here, guys. All right. Now you still have a week to prepare for your test, for your quiz. Okay. I'll tell you what's on the quiz. Right. Your quiz will have 2 short answer questions. Okay. So these are questions where you have to type the answer in the box. Okay. I will let you know both of these short answer questions will be based on lecture 1. Okay, the scientific method stuff, and it will be things that we've done. Okay, it's something that we've talked about that we did an exercise on. So if you need to rewatch lecture 1, you can find the recordings. Okay, there will be 17 multiple choice questions. Okay, with single answer to them, right? Just one correct answer. 17, and then there will be 5 multi-select questions. Okay, 5 multi-select question. That means they have more than one possible correct answers. Okay, the quiz will be 45 minutes long. That should be plenty of time. Do we have any questions about the quiz?
We're using the lockdown system as well?
The lockdown browser, you mean?
Yeah.
Yeah, that's correct. Okay, so if you haven't already done that, please try it out. And for those of you who don't know, the lockdown browser will lock your computer. You will not be able to pull up any files. Okay. So what I mean is, like, if you want to have notes, and you are allowed to have notes, right? Like, you need to have physical copy of the notes. Okay. Like, you won't be able to pull it up on your computer. Right. So print
Are
it
we allowed
out.
to have
Write it
notes?
out. Yeah, you can have notes. Okay. You just can't access it through your computer. Okay. So what
Okay,
I've been
so we can
telling
have our books.
yeah, you can have your books, sure.
Oh,
Yeah.
cool.
So what I'm saying is, like don't flip through the book to find the answer. Like, you won't have time. You only have 45 minutes, right? Okay. Like, make a study sheet. Okay. Have a cheat sheet, summary of key things. Like, if you cannot remember, phospholipid is glycerol, 2 fatty acids, and 1 phosphate group. Write it down. Okay. In the process of making the cheat sheet, it's a part of studying, right? Okay. So I just want to make that clear for you. And this is only for my class, okay? I don't know what your other profs are telling you, okay? Maybe they don't want you to use your notes. That's, you got to follow their rules, okay? But for my class, it's fine. For people with extra time, okay. So if you need accommodation, right? Some of you might have an IEP, right? Like an individualized education plan, right, from high school. So how it works is you have to go and talk to the people at CALS, okay? CALS. You just look it up on on Centennial, okay? Like, you type in your Google search, and then you type in CALS. This is the Center for Accessible Learning and Services, okay? At the college. Right? So, like, you need to talk to those people. They would give you the proper documentation, which you will then forward to me, and then like, you have to send me the document, right? And then I will give you the extra time. Okay, but without the documentation, I cannot do that yet. All right. So yeah, send me an email if you want to learn more about it. Yeah. Anything else about the quiz? It's all of lecture 1 and all of lecture 2. Okay. Yes, Georgia, go ahead. And then Ramon.
I was gonna ask you
Yeah.
after class.
Oh, sure. Yeah. Ask me
Okay.
after class then.
Thank you.
Mhm.
Are the questions in order, like the 17 questions and then the 5 other questions as well?
Oh,
Or-
no, yeah, they are all randomized. Okay. Yeah,
Alright.
but you will know it's a multi-select because the question will say choose all or select all correct answers. That's how you can tell.
All right, thank you, sir.
Yep. Okay, I think it's a pretty reasonable test. It shouldn't be too hard. If you study, we'll see how it goes. All right, why don't we take a 5-minute break, and we will continue when we come back. Let's keep going. Next macromolecule we will look at is proteins. This is probably the hardest one to understand. All right, so if I'm going too fast, please just stop me. And you know, if you're confused, ask questions. So just like before, when we learn a macromolecule, we want to know the main atom that makes it up, right? So we have the carbon, the hydrogen, the oxygen. Those are the triplets that always exist, okay, regardless of which macromolecules we're talking about. And for proteins, we also have nitrogen in it and trace of sulfur. Trace means just a little bit, okay? Just a little bit. The monomer of protein, the building block of protein, is something called amino acids. Amino acids. And there are 20 amino acids in total. So right away, it's much more complicated, right? Remember, in carbohydrates, there were only 3: glucose, galactose, fructose, right? But in protein, there are 20 in total. You don't have to memorize any of them, okay? Just know that there are 20. And out of the 20 amino acids, 9 of them are essential. Right? So it's the same word again, right? Essential, right? In the context of what we're talking about, essential means your body cannot make it, and you need to get it from your diet. So here are the 9 essential amino acids. Again, you don't have to memorize them, okay? I'm just showing you in case you're wondering. So, you know, we have something called like threonine, which you can get from eating things like watercress. I don't even know what this thing is. Pumpkin, chia seed, okay? Phenylalanine, you can get it from like avocado beans, so on and so forth, okay? But generally speaking, if you're not a picky eater, you should be able to get your essential amino acid just from your normal diet. Okay? There's no need to like go out of the way to you know, eat more of a certain kind of food. Now, one of the essential amino acid here is phenylalanine. Okay, phenylalanine. Some people are born with a rare genetic conditions in which they cannot break down phenylalanine. Okay, so there's this recurring theme that I have been showing you, right? Like to break down things, you need to have a special enzyme for it, right? And you know, earlier we said you don't have an enzyme to break down lactose, then you become lactose intolerant. If you don't have the enzyme to break down trans fat, then they built up in your body, right? And here for these individuals, they don't have the enzyme to break down phenylalanine. And since phenylalanine is very common in many different types of proteins, including like milk, like breast milk, for example, the phenylalanine is going to build up in the body, and it's going to lead to brain development problems. So that's the first thing that they will test in a newborn baby. Okay, they will do a what's called like a heel prick, which is what is shown here. They take a little bit of blood from the baby's very cute foot right here, and then they will run tests on it. And if they find out the baby has the condition, which is called PKU for short, okay, phenylketonuria, then they have to go on a special diet. They can't drink the breast milk. Okay, they cannot use regular formulas. Okay, they have to have a special formula where they don't they exclude the phenylalanine in the mix. Does that make sense? Okay, so that's a genetic condition. All right, back to the monomers of proteins. They are called amino acids, right? There are 20 different amino acids, but they all share the same core structure, right? For example, they all have a central carbon right here. Central carbon. They all have a hydrogen. And here, this part here, the NH2 here, this is called the amino group. amino group. And over here, this is called the carboxyl group, okay, which is an acid. That's why it is called an amino acid, because it has both. So all the amino acids, all 20 of them, will have these core structures. Okay, the central carbon, the amino group, the carboxyl group, the hydrogen. What makes them different is the R group right here. This is called the side chain. And there are 20 different kinds of side chains that exist, creating the 20 different amino acids. Okay, I'll show you on the next slide what it looks like. Now, I know it's very small and really hard to see, and this is not exactly what it looked like around the other way. Here, maybe I have a better picture here. Oh, there we go. Okay, so let's take a look at this one instead. It's the same kind of information. So look, all of them has this central carbon, you know, the carboxyl group, the amino group, right? All of them has that. And here are the different side chain, right? CH3, right? They all look different. There is the sulfur that I was talking about, right? So the different side chain is going to give them different properties. Some of them are non-polar. Some of them are polar, right? Some of them have positive charge. Some of them has negative charge. And it just gives them different properties. That's all you have to know. All right. So the R, the side chain, is unique for each amino acid. Okay. And it gives each one different property. Okay. That's all. Now, how do we connect two amino acids together? By now, you should be familiar with the process. We've learned it so many times already. We will be using dehydration synthesis. Okay, so we will remove an OH, from carboxyl group, and then we will remove H from amino group. And when you do that, you get your water molecule, right? You get your H2O. And you form a bond that connects them together. This bond is called a peptide bond. Right? So now you have a dipeptide. It's the same recurring concept, right? Like if you put two sugar together, you have a disaccharide. If you put two amino acids together, you have a dipeptide.
So, sir, we could say the C and the N bond them together?
Exactly. There's a glue, right? Sharing of electron between the C and the N. Now we will look at how proteins fold up and form a particular shape, right? Let me see. I should tell you right now that the shape, the structure, okay, the shape of a protein is related to its function. That's a key thing you have to remember. It's a recurring theme that is going to continue to show up. Okay. What was written on the last slide? Oh, here. Maybe you can take a quick picture and then we'll move on. All right. So the shape of the protein will basically determine its function. Or you can say the function is dependent on the shape. It's like a key and lock relationship. Each protein has a unique shape. Each protein it's like a key. And the key is only going to open like one specific lock, right? If you bend the key or if you like, scrub off the grooves, it won't open the lock anymore, right? And that's exactly what happens. We need the protein to have a particular shape. If you change the shape, and it could happen, then it loses its function or it alters its function. Right, so now we're going to spend the next couple of slides looking at how the protein folds up to create its shape. All right. So we have 4 levels of structures: primary, secondary, tertiary, and sometimes we have the 4th level, which is called quaternary. So let's start with the primary. The primary structure of a protein is the unique sequence in the polypeptide. Okay. So I know there's a lot of terms, like I know, like polypeptide just means the whole thing, just like the term polysaccharide, right? It's just really, really long. Now these balls, right? Those are amino acids. And remember, you have 20 amino acids, right? So imagine having a tray of beads with 20 different colors. And now I ask you to build your necklace. All right. So the order of the color that you use, that would be like the primary structure of the protein. Like, is it red, blue, green, green, yellow, red, blue, purple, purple, orange? Like if you read it one by one, the sequence, that is the primary structure. Okay, except we're not talking about like colors, right? In the case of protein, we're talking about the amino acids. And let me just go back here to show you. There are 20 amino acids. So which one are we connecting together? Are we using like alanine connected to, tyrosine, and then maybe another tyrosine, and then we will put a proline, and then a histidine? Like, what do we put together? What's the sequence? What's the order? That is the primary structure. Okay? What determines the order, what determines the sequence, the primary structure of your protein is your DNA. Okay? We will learn more about this relationship later on in the course, but that's what your DNA does. It tells you how to build protein. Okay? You look at the DNA and then you have the instruction to make the protein. So that's the primary sequence. Okay? Primary sequence. Give me a second. I just need to grab some props. All right, so here, this twist tie, right? That pipe cleaner, whatever you want to call it. This is the primary sequence, right? From one end to another end. And now what's going to happen is some part of this is actually going to wound up, and you're going to have this, little spiral here, and some part of it is going to, go up and down and form these zigzag pattern. Okay, so whether it forms the spiral or whether it forms the zigzag, that is the secondary structure. Okay, some part of it is going to form the coil, which is called alpha helix. Some part is going to form the zigzag, and that's called beta pleated sheets. Okay, and whether it forms the coil or the zigzag will depend on the sequence. Okay, some amino acids will have a higher chance of forming the helix. Some will form the beta sheets. So in any protein, there would be some alpha right? And then there might be some beta sheets. Okay, it could be only coils, maybe, or it could be just the beta sheets, or it could be a combination of both. Okay, you don't have to have both. That's what I'm trying to say. Okay, you could only have one type, or you can have both. That's the secondary structure. Do you guys understand? Primary is the sequence, the order. Secondary is whether it coils up to form alpha helix, or go up and down and forms the zigzag, the beta sheets. Okay, so this is a way that the scientists use to represent it. The arrow that represents the beta sheets, and then the ribbon you can see at the background, right? That represent the alpha helix. Beta sheets. All right. And you're like, oh, who cares whether it's alpha or beta? Like, why are we learning this, right? So let me give you one relevant example. Okay. So mad cow disease. Do you guys know mad cow disease? How do you catch mad cow disease, huh? Anybody know? Okay, food, yeah, food is one way to catch it. Eating contaminated beef, right? Okay, so for those of you who don't know, mad cow disease, as the name suggests, comes from cow, right? But it could cross into human as well. And it's not caused by virus, it's not caused by bacteria, which means you can cook the food completely, and if the food is contaminated with the mad cow disease, cooking it is not going to get it away. You can still catch it. All right. So when a person catch mad cow diseases, their brain is going to have holes in it. And this is a picture of the brain of someone who died from mad cow disease. You can see all these empty space, all these holes. Okay, so terrible thing to get, right? So how does that relate to what we're talking about? Okay, long story short, in the body, in the brain, we have a protein called the PRP protein. Okay, PRP protein. The normal version is going to be called PRPC, which has only alpha helix here, right? You see alpha helix? Okay, so just alpha helix. Someone asked, will this be on the quiz? I honestly don't remember. Maybe. Over here, for cows that have the mad cow condition, their normal protein, their normal PRPC changes its folding, so that instead of having the alpha helix, right, this is the disease one. This is the prion form, right? That has just beta. Okay, and that's the bad one. This is the bad one. Okay, this is the normal one. Right? So this one is called the mad cow form. And what happens is, oh, sorry, there's no next slide. What happens is this, right? If you eat a little bit of contaminated proteins, all right, that causes the mad cow disease, the misfolded protein will go bind to the normal protein in your body, and it will cause that one to change and misfold as well. Okay, so let's say this is the normal one, this is the bad one. The bad one will come, and then now you have two bad ones, and then they will go bind to another two good ones and change them into bad ones. And before you know it, a majority of your normal proteins will be misfolded, okay, and they start to come together, and it causes your neurons to die, and then it leaves holes in your brain, and that's mad cow disease. Does that make sense? Okay, so this has to do with the secondary structure. All right, like I said, if you change the shape, you change the function, right? And sometimes changing the function is going to cause major problem in the body. All right, so we have the secondary. Secondary is like this, right? With the spiral and then the zigzag. Okay, I don't know if you can see, this is up and down, and this is actually spiral, right? Now the next level is tertiary. Okay, tertiary means this thing is now gonna fold up, into a ball. into a ball. So like I have a 3-dimensional structure. The 3-dimensional structure is the tertiary structure, okay? And what holds the 3D shape together is the interaction between the side chains. Remember, each amino acid has a different side chain, right? So how the side chain interacts together is going to help determine the overall 3-dimensional shape. Okay, so here are some examples of how side chains interact. Okay, so you can have some that comes together, right? To hydrophobic things tend to come together. Okay, you might have an ionic bond. You might have hydrogen bond. Okay, so all these things help stabilize it. You don't have to memorize any of these. Okay, the key thing to know is that the tertiary structure is stabilized by interaction between the side chains. So let me switch to the study guide here, and let me help you put it together in. And you should do all these, right? I wanted to remind you, right? So this is question number 17. All right. First one, primary structure. That is the sequence of amino acid, right? And it's determined by your DNA. What holds them together? That is the peptide bond, right? Peptide bond is like a type of covalent bond, right? It's a sharing of electron. So illustration, that's just a bunch of amino acids connected together. Right? I hope it makes sense so far for you. What about secondary structure? Right? What is the secondary structure? It means it's either forming the alpha helix or what's the other one called? Beta sheets, right? I know it says beta pleated sheets, but I'm just gonna put beta sheets here to help you, keep it simple for now. And bonding characteristics, what's holding them together? It is the hydrogen bond of the backbone. Okay, let me show you the backbone again. Right? The yellow part here, right? That's the backbone. The backbone doesn't change, right? It's always like the same thing. C double bond O, and so on and so forth. I can draw this out for you in a bit to clarify it. Okay. So illustration, right? You either have these helix or you have the zigzag, right? Tertiary, right? Tertiary. This is talking about the 3D shape. 3D shape of the protein. And what holds them together is the interaction between side chains, the R group, right? Interaction between the side chains. And so now you will have an overall 3D shape. It's, you can't really draw 3D here, but, it will fold up into a ball. Okay. You still have the, zigzag and the helix, but now it's like, into a ball. Okay. Now I'm gonna quickly show you what I mean by the backbone. All right. I mean, there's a question about it later. Earlier. But I will show you what it looks like. Okay, I'll show it to you like here. Okay, draw the peptide backbone. You don't have to copy. All right, this is probably at the 1 hour and 1 hour and 30-minute mark, maybe, if you need to watch this again later on. All right, just watch. I'm not gonna ask you to draw it on the test, but I just want to show you what it means so you understand. Look, the amino acid look like this, right? I told you. The amino acid for all of them looks like this. And what makes it different from one amino acid to the other is the R group, right? So that would be like R group number 1. And let's say I put another amino acid here. Okay, just watch. It will make sense, I promise. Okay, so this is another amino acid. R2 now. Okay, and I'm going to put a third amino acid here. Okay, now watch. This is the amino group, and this is the carboxyl group, right? And then here again, we have the amino group, and then the carboxyl group, and then the amino group, and the carboxyl group. Right? It's just repeating itself, right? Okay. So how do I bond them together? All I have to do is remove. No, you don't have to remember the structure, okay? Like you don't have to draw anything on the test, right? Okay, but this helps you understand it. Okay, watch. I can remove the OH and the H, right? And then I would connect them. Okay, someone said earlier, I think it was Georgia, said, so the C and the N is what forms the bond. That's right. And here I can remove the OH and the H, and then I form a bond. Right? And then I can do it again for the next one, right? Okay. And you can see, if you keep on doing this, the backbone remains unchanged. Right? What's the backbone? This whole part here is the backbone. Okay. Look, it begins with N and then CH, CO, N. CH, CO, N. CH, CO, and then if I connect another one, it would just be another N, right? So if I go back to the PowerPoint here, you can see, right? CH, CO, N. CH, CO, N. CH, CO, N. And then it just keeps on going. So that's what this yellow ribbon is. That's the backbone. Okay, that's the backbone. So when they ask you in the chart, what holds the helix and sheets together is the hydrogen bonds of the backbone. That's all. Any question so far? I know it's a lot of information. I'm sorry.
Sir, like for the one that you just show us up, I think that's 15.
Yeah.
Yes. If we see this question on the test, how would we illustrate this to you? Because
you won't see this question on the test because I won't ask you to draw anything. Yeah.
Oh, thank God.
Yeah, what a relief, right? Yeah.
Right?
No, like the questions are really reasonable, guys. Okay.
I'm thinking,
like,
how am I gonna draw this?
Yeah. You don't have to draw anything. Okay. Like I think if you study the question in the study guide, you should be fine for the quiz. Okay. I don't want you to think I'm not crazy. Okay. I'm not trying to fail anybody. Okay. Finally, quaternary structure. Not all proteins will have a quaternary structure. Some will. Okay, some will. And what that means is you would have multiple peptides coming together. Okay, like I don't have another one of these, but like imagine this is one of the peptide, and then you will have another one that come together, and then another one, like this picture. Okay, so this protein has 4 smaller proteins in it. Okay, that's the quaternary structure. Okay, so hemoglobin, which helps carry oxygen in the body, that's an example. Right? It has 4 different parts to it, and in order for it to function properly, I need all 4 parts, not just one, not just 3, all of them. Okay, so that's the quaternary structure, and these are held together by interactions between the peptides. So there you have it. This is the
Quick question.
Yep, go ahead.
For on the quiz, you know how we did that sheet with the practice sheet with Reedy and posterior and all the theories and stuff?
With the anterior and posterior, what do you mean?
Yeah, the one that we had to practice on.
The practice quiz?
I don't think that was a quiz. I think that was just a practice sheet. I'm not sure.
Practice
The
sheet.
worksheet. I think I'm already in the process of doing that.
Okay.
Would that be an example of a cheat sheet? Like if I copy out, if I print out that whole sheet?
I am not sure which sheet you are referring to. Could you explain that to me again, please?
It's on the assignment page.
You mean the study guide?
The study guide, the one that we have to answer all the questions and fill them in.
Oh yeah, like this one, right? Like this study guide, right? Is that what you're talking about?
Yeah, but
Like this one?
yes.
Yeah,
This
so
one.
that yeah, sure, that could be a cheat sheet if you want, right?
But this will be on the quiz, right?
Yeah, but like in multiple choice format though,
Yes.
right?
Oh.
Right. So like I was saying earlier, right? For those of you who missed this, right? I just want to be very clear. I'll post this again. The quiz will have 2 short answer questions, okay? And they will be based on lecture 1. Now I want to be clear. The short answer questions will not be exactly same as these one, because then what's the point, right? Like I know you would just be able to pull this up and then you type in the answer. That's not really the point of a quiz, right?
Okay.
It would be, but it would be a question that's based on what you've learned here. Okay. I don't know if that makes sense. And then you will have the multiple choice questions, 17 of them, and then 5 multi-select question.
Cool.
Okay.
Thank you.
Yeah, no problem. All right. How many more things do we have? Okay. I don't think we can finish the DNA today. We'll just finish the proteins and then we'll finish the DNA next class. Okay. DNA is really short. You should be able to learn it, on Tuesday and get tested on it on Thursday. Shouldn't be a problem. Okay. Importance of protein structure, right? I already told you the shape determines its function. If you mess up the shape, the function will be wrong. Okay. So there is a fancy word that describes the shape of a protein, and that is called conformation. Conformation. Okay. Now, when you heat up the protein, for example, you will change the protein structure. The heat will destroy its structure. Right? And when you break down the protein structure, we say it has been denatured. Okay. So if you think of an egg, right? The protein, which is the egg white, okay, many people don't know that the yolk is where the lipids are, but the protein is in the white. And when you cook the egg, it changes from transparent to like white in color, right? And that's irreversible. You cannot uncook an egg. So what in that process, the heat basically denatured the protein. It unfolds the protein, it breaks down the protein to a certain extent, and it becomes like this. Same thing for cooking the meat, right? When the meat changes from like red to brown, you are essentially denaturing the protein in it. So there are various factors that could affect the shape of the protein, that could break it down. Right? Temperature is one. You can change the pH, right? The acidity of the environment, that could unfold the protein, which is why we have to keep the blood pH at a very narrow range. If you go too high or too low, it will cause proteins in the bloodstream to denature, to unfold, and they lose their functions, and that could be deadly. Changing the salt concentration, changing the type of solution that it is in will also cause it to misfold. If you change it from, let's say, a hydrophilic solution to a hydrophobic solution, it will cause the protein to flip inside out, and that is going to affect its function. Here is one example. Right? Remember, the 3D shape is determined by the sequence of the amino acid, right? So if you change the primary sequence, there is a chance that you will change the shape of the protein. And the classic example that every teacher would bring up at this point is something called sickle cell anemia. Sickle cell anemia, anemia means you have shortness of breath, you cannot carry oxygen effectively in the body. That's what it means, okay? So as I told you, hemoglobin, right here, this word hemoglobin is a protein that is in your blood cells that helps you carry oxygen. Okay, and it has 4 separate chains to it. All right. So here are the first 7 amino acid in hemoglobin. It has a 146 in total, but here are the first 7, okay? Valine, histidine, leucine, threonine, protein, glutamic acid. That's what they stand for. You don't have to know it. Now, for sickle cell anemia, because there is a mutation, a change in the person's DNA, and the DNA has the instruction to make the protein, right? So if you change the instruction, you're gonna change the sequence. And number 6 here, instead of having a glutamic acid, now it becomes a different amino acid. It becomes VAL, which stands for valine. And just that single switch in one of the 146 amino acid will cause the hemoglobin to misfold. When the hemoglobin misfolds, it will change the shape of your red blood cell. A normal red blood cell looks like this. It looks like one of those, looks like a donut, okay? Except the middle is not completely through. It's like a jelly-filled donut, and you kind of pinch it a little bit in the center, right? But when you misfold the hemoglobin, you will cause the cell to become moon shape, sickle shape. Okay, so that's called sickle cell anemia. These red cells will get trapped in small blood vessels, and then they will die off and not be able to carry oxygen for you. Okay, so that's an example of where if you change the primary sequence, you end up with a different 3D shape, and then you lose the function. So what are the functions of proteins, right? Like we haven't really talked about that. Yeah. So a lot of structures in the body is primarily proteins. Okay. Muscle is a lot of proteins in it. That's why people drink like powder shakes, right? Protein shakes to bulk up, right? Things like nails, horns. I mean, we don't have horns, but like animals, they do. Hair, spider webs, all these are proteins. Earlier, we talked about hormones. Some hormones are steroid hormones, like the sex hormone, testosterone, progesterone. Those are steroid hormones. Other hormones in the body are protein hormones, okay? Like insulin. You probably have heard of that. It helps to regulate blood sugar. That's a protein hormone. So you can imagine if someone has a mutation in the DNA, and that instruction allows the body to make insulin, and now you mutate it, you can no longer make insulin, right? And that could cause you to become diabetic. We also use protein as an energy source. It's not the preferred source of energy, right? We much prefer carbohydrates, but we have proteins in our diet. We can burn them and produce energy as well. And lastly, there is something called enzymes. Okay? I've been trying to introduce this concept to you throughout the lecture already, right? These enzymes are proteins that have a specific function to like break things down, maybe, right? To carry out chemical reactions. So a brief word about enzyme. Enzymes are proteins that catalyze, which is a fancy word for speeding up chemical reactions in the body. Okay, so here's a graph. Okay, don't be intimidated by it. It's very straightforward. Okay, so this is the reactant, the starting material of a reaction, and then you want to get to the products. In order for you to do this, you have to supply a little bit of energy. Okay, so the energy that's required, the activation energy that's required for a reaction to occur, is usually quite high. It's so high that it would be impossible for you to do in the body. Right? So that's what the enzyme is for. The enzyme is going to lower the energy requirement. You can still do the reaction, but now you don't have to do as much. Think of it as like a scholarship, okay? So expensive to go to study at a college, study at university. You have to pay like, $10,000. But with the help of scholarship, right? You no longer have to pay $10,000. You only have to pay like, I don't know, 7,000 or something like that. So instead of supplying this much energy to do a reaction with the help of an enzyme, you only have to have this much, right? So you can do it within the body. That's basically what it is. Okay. So this is how an enzyme works. An enzyme has a particular shape. Think of it as like having a slot to fit things in. Right? The slot that you fit things in is called an active site. Okay, the slot to fit things in. And the thing that you want to fit in it is called the substrate. Okay, the thing to put in the slot. All right. So here is an example. And let's say this red cube, let's say that represent glucose and that represent fructose. Okay. If you try to connect glucose and fructose together, you would have to supply this much energy. Okay, which is not possible to do inside the body. So what if we want to put them together inside the body? Well, we need to lower that energy, right? We need a scholarship, right, so to speak. So that's where the enzyme comes in. Right? So these substrate, the reactants, will fit in the active site, and then the enzyme will help them do the reaction, and then it will split out the product. So in this case, if you pick glucose and then fructose and you put them together, you end up getting sucrose, right? So that's a reaction. Similarly, if we start with sucrose, you can fit it into the active site of a different enzyme. I know they look the same here, but it has to be a different enzyme. And then the enzyme will break the bond for you and then split out the product. Okay? Almost all chemical reactions in the body will require some sort of enzyme to do it. Okay? And all these enzymes in the body are made up of protein. Okay? So the person who was lactose intolerant, they don't have the enzyme to break down the lactose. Why not? Well, because there is a mutation in their DNA. Okay? The instruction on how to make the lactase is missing or it's incorrect. So they don't have the enzyme. So they cannot break it down. Okay? That's basically what enzymes are. Last slide. Sources of protein. Where do we get protein? It's very straightforward, right? You just eat the meat and you would have protein. If you are a vegetarian or vegan and you don't eat meat, you can get it from from vegetable source, right? So things like beans, soy product like tofu, nuts, lentils, they are packed with proteins. Okay, so you need protein though. You cannot survive without protein, right? Because you need the raw material from the protein to make the proteins that you need, right? Think of the insulin. That's a protein that you need. You can't just drink insulin, or it doesn't work like that, right? You need to eat protein, break it down into amino acids, and use those amino acids to build the insulin. Okay. Here are a few questions, which we don't have time to go through now. You're more than welcome to try it on your own. We will take it up first thing in the next class. Okay, and then we'll finish up with the DNA. It's not too long. Okay, we should be able to finish this in the first half an hour of next class. And for next class, we will also start on lecture 3. Okay. There's a couple other things I wanted to tell you about the quiz. Oh yeah, so like on the day of the quiz, right, which is next Thursday, we still have class, right? You still have to come to the regular class, learn the lesson, and then you can do the quiz on your own. The quiz will be available from 6 a.m. all the way to midnight, right? So you can do it between my class and math class if you want, or you can do it, in the evening, right? Study more first, or you can do it first thing when you wake up, okay? And here is one more thing I want to tell you about the quiz, okay? Let me just stop the recording here. Stop the recording.